Great. Hello, everyone. It's my pleasure to convene today's meeting of the Semantic Technologies Committee. I'd like to introduce quite briefly Jianyang Gu at Ohio State University. It's I have not met personally, but I've been keeping track of your work now for a few months since Tanya introduced us. So it's very exciting to have you talk. I'll go ahead and let you introduce yourself in more detail and provide an overview of your work. Thanks for joining us today.
Thank you very much. So I'm Jianyang Gu. I'm a postdoc at the Ohio State University in the Department of Computer Science and Engineering, with the Imagomics Institute. So my research is mainly using data-centric AI to build interpretable and scalable models for scientific discovery. So I'm going to share my screen.
Should be able to now.
Yep. So can you see my screen okay?
Yes.
Okay, cool. So yeah, today I'd like to talk about from pixels to planet, how to build scalable and grounded AI for science. Um, so I'll get started. First of all, we know that AI has demonstrated a lot of new possibilities. Like every day, we can use AI to help us draft emails, to edit photos, and to generate videos. And I know, for example, in computer science, a lot of students they have been using AI agents to write coding assignments. So really, like AI has enabled a lot of possibilities. And the effectiveness of AI models is largely built upon this machine learning pipeline. So where data, we can treat it as, for example, images. They are fed into the machine learning models to get representations, and then the model is going to produce predictions. So in mainstream benchmarks and general purpose tasks, this pipeline works perfectly because the settings are relatively controlled. So usually the benchmarks, for example, image classification, they have well-defined labels, and evaluation is usually a single metric, for example, classification accuracy. So with the well-defined labels and fixed metric evaluation, this pipeline works pretty well. However, the problem happens when we try to apply this pipeline to interaction with the world and for AI and use AI for science. Because scientific domains, they are often not about closed set decisions. Scientists, they want to discover new categories, they want to propose new hypotheses. And then we will need to rethink this pipeline to make AI work for science. So in this talk, I would like to use biodiversity as a motivating domain. It's mainly because biodiversity has relatively well-structured data and is closely associated with machine learning and computer vision. So for example, here I show the pipeline of an image classification task where an image is passed through a neural network to produce the label penguin. So this is a very classic and foundational like object classification task. And biodiversity has contributed many like very early data sets into the domain of image classification, such as cup, and birds, pets, flowers. It covers a lot of different species, like plants, animals, a lot of them. And more recently, there are also some more challenging tasks. For example, we want to detect animals, we want to detect this wildlife, and then we want to understand what their behavior are. So, so this is really a domain that has been like connected to machine learning. And if we look back at this pipeline, where data is passed through machine learning models to generate representation and prediction, so beyond benchmarks, if we want to advance scientific discovery for biodiversity, the first requirement is to get a very comprehensive understanding of global biodiversity. So if we have this target, then what would be the problem when we work toward it? So the first problem we identified happens at the left end, and it's the data. So iNaturalist 2021 is a dataset that has been well incorporated in the field of computer vision. Like people love this dataset because it provides very fine-grained setting and it's relatively large because ImageNet has 1 million images while iNaturalist has 2.7 million. It's much larger than ImageNet and also it has 10 times more categories. However, these 10,000 species, if we consider, okay, how many species are there on this earth? So roughly, we have about 2 million species that have been documented. And further, if we consider all the possible species on this earth, so this is not like some definite number, but there are multiple guesses, but one estimation is there are 8.7 million species. So in this sense, we can know that iNaturalist, these 10,000 species, is only a tiny, tiny bit among these 8.7 million. So this data set, while it is well adopted by computer scientists, cannot really represent this Earth. And what is worse is that if we look at the Global Biodiversity Information Facility, GBIF, it provides a very comprehensive coverage of biodiversity data. And we can see it provides detailed metadata, including species, time, location. So we know when the image is captured and where it is captured. And we also know, like very detailedly, its species label. However, scientists they want to know more. They don't want to only know like what species it is, the time and location. There are many more questions. For example, what is it doing? What does it look like? What life stage is it at? And is it male or female? There are so many more questions that cannot be simply answered by species, time, and location. So in this sense, the data scarcity is really limiting like our global understanding. Like suppose we have a machine learning model, it's really restricting its understanding of this global biodiversity. And the second problem happens at the other end when the model is producing a prediction, and especially when the prediction is presented to a scientist. So suppose the model pass through this image and produce a prediction of tail duck. So scientists, they might be curious about what evidence the model is using. So we would want an explanation of why the model thinks it is a tail duck. So this why explanation is necessary because they let us verify whether the prediction is supported by some biologically plausible evidence, for example, the patterns on the head, rather than the background environment. And more importantly, if we consistently identify a pattern, like a visual pattern across samples of the specific species, the pattern can potentially be treated as an explicit trait. So some of these traits may still be unknown. Like, of course, like scientists and people, we have recorded a large number of traits for different species, but some traits, they might be hard to describe. They are predictive and consistent, but they might not have been well documented by humans. And that's where explanations can help us discover new concepts and new traits. So even if the explanations are critical for scientific domains, existing models rarely produce faithful and reliable explanations along with the predictions, which is the second issue of this pipeline. And these two issues are not unique to biodiversity. They also restrict many other scientific domains such as remote sensing and drug discovery, where the collection of real-world data is costly and difficult to cover all the data distributions. On the other hand, scientific domains also require interpretability to facilitate high-stakes decisions and understanding of failure modes. So in this talk, I'm going to introduce how my research solve these two challenges from scaling and creating data as well as interpretable and explainable modeling. So firstly, I will start by scaling data. So this is a work published in last year's NeurIPS, and it's a work with Tanya Bergwolf, William Chow, and Yu Su. So this starts from a very simple motivation. So suppose today we want to train a species classification network for birds. So now we are using a data set, a very famous data set called CUP. So it has a bunch of bird species, and now we train a network that can predict very well on this data set. Okay, tomorrow I have new idea. Okay, I want to test this model on some other species. There's another bird data set called NA birds. But of course, this network is not going to work because they are trained on different species. Okay, then simple, I can train another network. But this will lead to an infinite number of models. So because we want to know more, like not only birds, we also want to know pets, we also want to know flowers, and then we will train a lot of models. Then the question comes when we want to really use the model, like when there is an image and we want to know which species it is. The question comes, which model should I use? Right? So we don't exactly know which model may cover these species. And beyond species classification, we also want to know like what is it doing, what does it look like? There are a bunch of these various questions that we want to know, which cannot be answered by like any of these models. So then we will have really like infinite models, and that is really a headache for scientists. So that's the motivation of why we want to develop a foundation model. So we want a model built upon the whole tree of life, like it covers a large number of species. And we not only want this model to do species classification, we also want it to have like better ecological understanding. For example, we want this model to do habitat classification. We want it to detect different diseases. And this really means that we want this foundation model to have an ecologically meaningful representation space. And the first step, of course, is to curate this data set. So we work, I led a team of about 20 people, and we work with domain experts. We curate data from four sources: GBIF, FathomNet, Encyclopedia of Life, and Bioscan. So we develop a series of tools, including downloading these data from different cloud servers. We align the taxonomic labels across these sources, and we process the data to get rid of some data that we do not want. For example, there are some images containing human faces, and we excluded those images from our training. So we publish all these tools online. They can be publicly accessed. They're distributed downloader, text on the Pi, and Tree of Life toolbox. So these tools they provide a tool like a solution for new data set. So they are not only used to reproduce our data set, but can also be treated as a pipeline. Like when you collected a new data set, or if you have some new images, then how do you transfer them to machine learning ready? So with these tools, we collect Tree of Life 200 million. Um, so this is a data set with more than 214 million images, and it covers 952,000 taxa. So that's almost a half of the species we know on this earth. And with this huge data set, um, we want to know, okay, now we have the images, and the next step is how do we inject the domain knowledge? Again, our target is ecologically meaningful representation space. And in that sense, we want to leverage the taxonomic labels to inject this domain knowledge. So taxonomic labels, they not only indicate species names, they also reflect, in some sense, the ecological similarity between different species. In this example, we have honeybee, bumblebee, and yellow jacket. So honeybee and bumblebee, they belong to the same family, while yellow jacket is in another family from the same order. So in some sense, they are all similar, but honeybee and bumblebee, they are closer in the taxonomic distance, and also in their ecological roles, they are more of pollinators, and in comparison, yellow jacket is more of a predator. So while this relationship does not always stand for all the taxonomy, but it provides a valuable prior in understanding the ecological function and the ecological roles of different species. So how are we going to encode the taxonomic information into the models? So we used contrastive learning with these taxonomic labels. So here, in this example, we are maximizing the similarity between an image and the text embedding of its taxonomic label, while minimizing the similarity to other labels. So here we have two labels, and we are maximizing the similarity to, for example, label A and minimizing the similarity to label B. So in this sense, they are like really hard negatives. Um, and also, so although they are two different labels, but they in some sense they have shared the labels from kingdom to genus. So in some sense, they are also similar. So while we are pushing away these two species, we are still keeping it maybe closer compared with some other species. So in this way, we are organizing this whole embedding space, this whole representation space, and we can organize similar species together in the embedding space, and because their text embeddings are inherently close. So this objective was first proposed in the first version of BioClip, and here we demonstrate that it not only represents species similarity, but also the ecological similarity. Okay, so with these large data sets and these objective function, we now train BioClip 2, and and we first evaluate the model on species classification tasks. We evaluated across different species, including animals, plants, and fungi, etc. So we see that by using more training data, BioClip 2 illustrates significantly better species classification accuracy than BioClip. So the first version of BioClip was trained on 10 million data, and now we have 20 times more images. And also, if we look into specific categories, this new dataset provides 3 times more fungi species compared with BioClip, and it then boosts the performance from 40% to more than 80%. This really provides the coverage on less represented species, and we try to mitigate these bias toward those species that have been well represented. And more importantly, as we discussed before, we not only want this model to do species classification, we also test this model on a series of ecological tasks, including habitat classification, agricultural disease detection, attribute identification, and species discovery. So we show that BioClip 2 performs much better than other like very popular visual backbones. So here, Cyclip 2 and Dino V3, they are some go-to answers. They are off-the-shelf models that people will often use when they need to deal with visual data. However, here we show that by training on a large number of biological organismal images, the BioClip 2 is really like doing great on all kinds of visual tasks. So this superior performance really depends on the ecologically meaningful representation space of BioClip 2, and therefore we would like to look into the embedding space. So here in this example, there are a bunch of dolphins finches. So all different colors represent different species. And these different species, they have different beak size because they eat different things. And we show that BioClip2, so in the middle, this is a uh, embedding distribution figure where we extract the embeddings of all these images and compress them into a 2D space. So we can see, first of all, similar colors are clustered together. So that means we have decent species classification performance. And then more interestingly, we can see that from left to right, the beak size gradually increases. Of course, there are some other features like color or like the thickness or other attributes of the beaks, but generally the sizes is gradually increasing. So this information has never been seen during training, and BioClip to automatically accomplish this space kind of organization thanks to the large training scale. And we show that this embedding distribution can be aligned with ecological functions. And another interesting thing happens within each species. So after model training, we expect that different species will be well separated. As shown in this figure, we have three different birds and their embeddings are well separated. The species classification performance is very good. Of course, we can see there are one or two samples that are not classified correctly, but mostly they are well separated. But however, we know that many birds have different appearance between juvenile and adults or between male and female. So will these variants collapse into one single prototype after training? The answer is no. So here we have the darker images, the darker dots means juvenile images and the lighter ones are adult images. So we can show that they are even better separated compared with vanilla clip or the first version of BioClip. So how does this happen? We try to flip the distribution into a 3D space. So here we show that the juvenile and adults, these different variants, they are distributed in a, you can see it distributed very thinly along an axis. And this axis, uh, like it's not conflicted with species differences. You can see this gray platform represents like the prototypes of different species. And orthogonal to that platform distributes the variants, for example, juvenile and adult. So we have a formal proof in the paper to demonstrate that the variant preservation will not influence the species separation. And of course, I won't go into too much detail in this talk, but we demonstrate that by large scale training, this has been enabled, that the variance will not be diminished, and that is also why we can see that BioClip has very good performance on disease detection as well as attribute classification. So BioClip 2 has been adopted by a lot of stakeholders. For example, we know that the World Organization of Animal Health has applied BioClip 2 in their online model to classify different species. And the model has been downloaded for more than 300,000 times. And the dataset has also been downloaded for more than 60,000 times. And while we appreciate the rich ecological representation acquired in BioClip 2, we also want to know its use in real-world discovery. So here is a task where we apply BioClip 2 to discover new species. And this is a dataset that containing really new species that haven't even been named. So they definitely are not in the training of BioClip 2. And here we show some performance. So we are comparing BioClip 2 with the first version of BioClip, of course, and we are also comparing it with some frontier visual language models like GPT, Cloud, and Gemini. So firstly, we show that uh, classifying a species if that is known or novel, BioClip 2 has really like really high performance, almost 80% accuracy. Oh, sorry, it's F1 score of almost has 80% F1 score, and we noticed that GPT 5.2 it has very good performance on novel species detection as well, but at the same time, it performs really poor on non-species classification. So that means it genuinely just classify all the species to novel species, but it doesn't have a very comprehensive understanding on all the existing species that we know. And on the other hand, BioClip 2 does not only perform well on classifying if an image is a known or novel species, it can also roughly tell you what family it may belong to. So for the novel species, it has a almost a 60% F1 score in telling its family. That means given a new species, given like an image of potentially new species, BioClip 2 can very reliably tell you, provide some like highly possible family labels, and these will largely reduce the workload of scientists because they can mainly focus on the suggested family, the suggested families. So this tells us that this project of BioClip 2 tells us that structure and scaling can lead to domain-aware representations. So in our case, structure means the taxonomy and the contrastive training objective. And scaling means that we cover more data and we have larger capacities of our model. And then we can have the ecologically aligned embedding space and the space and also support discovery. So this is a transferable recipe to other domains as well, such as like medical and remote sensing. We can also adopt such a structure to help build like more domain-aware representation spaces. Okay, so BioCrypt 2 is good and it leverages these structured labels. But however, labels are not often like informative. For example, this fungi is named after Obama, but I won't say that it has any visual traits from Obama. And similarly, this frog is named after Darwin, but I also I won't say like they share any similar visual appearance. So this really means that some taxonomic labels, they themselves, they are not meaningful. They do not contain the necessary semantics that describe the visual appearance of the species. That means we really need descriptions and captions to help machine learning models acquire sufficient knowledge for these species. But sadly, even if, for example, a very comprehensive coverage from GBIF, we have species, we have time and location labels, but we don't have captions. There is no available descriptions on each image, and we know that it's super expensive. Even for human annotators, if we want humans to provide descriptive captions for each image, is very expensive. And like not all the people can do this job. We need experts. So that means generating all the captions by human is impossible. In this sense, I'm going to introduce the next work led by my student Ziheng. So this work has been published in this year's iClear, where we create these descriptive captions for different images. So we know that Wikipedia can be a really good source for these species descriptions. For example, this hummingbird, the Wikipedia has like very detailed visual information. However, we cannot directly use it in our training because these information are generic. They are not specific to each image. For example, this image is a female hummingbird, but if we directly use the visual information from Wikipedia, then we can like we have the risk of using the male descriptions. So we have a risk of matching the male descriptions to this female image, and that's not what we want. And another option, of course, is we can use large language models to generate captions, which like we agree that nowadays these AI agents, they are very strong. They have very comprehensive understandings of this world. But however, we know that language models, they can hallucinate. In this case, the language model says, oh, this hummingbird has a white throat and a bluish tinge along the tail, which are not very accurate. We can see there are some spots around this bird's neck. So, so the descriptions are maybe in some sense good, but they are not accurate. And we know that if the captions are not accurate, we are injecting wrong information, we are injecting wrong knowledge into a model. And again, that's not what we want. So what we did is we combined these two components. So we provide the Wikipedia visual information, and also we curate some format examples. So these examples will tell the model, for example, for birds, which are the traits that you want to include in the captions or in the descriptions. Because for different species, like we need to focus on different body parts, different traits. These format examples can tell the model like which traits you should focus to describe these species. And then we input this knowledge into the multimodal large language models, and then it can produce very faithful, like much more accurate. I won't say like 100% accurate, but much more accurate than without these context information. And then we train a new model called BioCap together with image with the species labels and these captions. And then we want to show that, of course, a BioCap, of course, is a better model, but we want to show like why it is better. So for this flower, in its training captions, there are two body parts. There are two parts like petal and anther. They are frequently mentioned in the captions. So petal and anther are these two parts in the flower, and we want to show that the model BioCap has a very accurate understanding of these concepts. So we try to make the model explain these two concepts. And we can see that clip model and bioclip model, they don't actually know what these two things are because these two concepts, they are probably not in their training data. And in comparison, BioCap has very accurately captured like where in the image trigger these two concepts. And these really kind of help us understand, okay, the performance improvement of BioCap does not come from some black box because we explicitly tell the model that you should look at petal and anther to predict these species. And that's how we make the model effective and at the same time interpretable. So similarly, we can use vocab to explain a lot of different biological concepts. Like for fish, flowers, frogs, we can you can see that it has very accurate highlight. It has very accurate explanation of all these different biological concepts. And further, it even understands behaviors. Like perch, when we try to use this model to explain perch, biocap will highlight the foot. And when we try to explain fly, it will highlight the wing. And when we try to explain feet, it will highlight the beak. So this model, by using these very detailed description, it has much better understanding of different biological concepts. And that's how we show that this model, its good performance comes from some interpretable concepts. It's like It'll be less black box for scientists because we know that it's more interpretable. And this is also a transferable recipe. There is a very broad application opportunity for leveraging domain knowledge beyond labels. So um, these can be extended to like other domains such as chemistry and geoscience. So we have another paper published in this year's CVPR where we leverage the descriptions on remote sensing models. So it also helps us to discriminate the data from different sources. So we know that remote sensing data across different data sources, they may look very different. So with descriptions, it provides us additional information beyond label. So that's how we make the model more accurate and more interpretable. Okay, now I've talked a lot about data scarcity, how we scale data and we generate new data that didn't exist to help us build a more comprehensive understanding of the global biodiversity. And the next step is explanation. So beyond training models, we're also designing methods to better explain these models, better explain their prediction and decisions. So here I would like to present a work also with students Hen and with Weiwen Chao. So this work has been published in last year's CVPR. It is mainly focusing on explaining model decisions. Okay, I will first start with an example. So you can have a guess of what is the trait of this bird. This is a blue grosbeak. So people can have a guess. What is the trait?
Probably the bright blue plumage.
Mm-hmm. Yeah, that's a good guess. Any other ones?
Um, oh
Beak.
my God.
Color on the wings.
Wings? Mm-hmm. Yeah, yeah. So there are like a lot of different traits on this bird. And now, how about this? So there is another bird, granddaughter. So then in this case, I think we will have a better sense of the trait, like how to discriminate between these two species, right? Because they both have blue feathers, they both have blue plumage. And their main differences are the wing patterns as well as their beak. So now there are some existing methods that can explain the model decision, which is called class activation map. So it tries to highlight like which pixels the model is using to produce a prediction. So here we have a trained neural network. We suppose that it has pretty high classification accuracy against these two different species. And now we want to explain the first image, that why it is a blue grosbeak. And the method will tell us, oh, it's mainly because of its blue, of course, right? It has blue in its name. Of course, the blue should be a very significant feature. But what if we ask the model to explain granddaughter? It's also blue. It's also blue feather. So, um, like now we are really getting confused because we are asking the model to explain the same image but with two different species, and it generates almost identical highlight maps. So this really raised the question: is it because the CAM, the class activation map cannot support fine-grained tasks? So we first look into how this tool works. So with an image, this is a bicultural image, and these are some like neural network blocks, and we can generate some feature maps. So of course, there are a lot of features. Like feature one can be the head pattern, feature two can be its belly, Feature 3 can be its wing. So there are a lot of features we can use to classify these image. And after getting these feature maps, the model will pass it through a linear classifier to produce logits. So these logits are the probability of different species. And suppose the probability for bicotille is 0.4. And now we backward this probability, we want to maximize. Okay, we say like we want to maximize this probability, then which feature map is important. So we will have a score based on the gradients. We will say, okay, the first feature map maybe contribute a lot to the final score, and the number two contributes a little less, and so on. So we will give a score for each feature map. And eventually we aggregate these different feature maps with a weighted average to get the final activation map. So it sounds perfect. And there have been, but however, there have been a lot of methods they are proposing to improve the GradCam. So there are some like newer signals like ScoreCam or some other different methods. They are trying to use different signals other than gradients. So the question is it really the problem of gradients, or is it really like lack of interaction between neural network layers? We say that the problem is not how it explains. It's not like which signal it is using, but what it explains. So here with the linear classifier, we can think that these classifiers, they are actually classification weights. So suppose we have a similar class, class D, which is teal duck. So it'll have also a similar like high probability because these two species are inherently very similar. So what does it mean by they are similar? That means their linear classifier weights are also similar. So because of this linear classifier, it can be treated as prototypes for different species. Therefore, the prototypes of bicotille and teal duck, they are inherently similar because they share a lot of features. And in that sense, if we explain one single species independently, it will inevitably uses a lot of features that are shared by other species. And of course, for the case of blue grosbeak, the blue feather is really a significant feature. So it will inevitably use the blue feather as the main explanation. And that's why we say the independent explanations can be ambiguous. Therefore, what we did is we explicitly create a contrast. We are not explaining a single species, but we are explaining why this image is class C instead of class D. So similarly, this is our target to maximize. We are trying to maximize this margin, and we are doing backward pass to to give scores to each feature maps, and then we aggregate these feature maps. So these are all the same steps as the very vanilla GradCam, and these can be treated as a spot the difference game. So we are trying to spot the difference between two species to create the explanation. And this is inherently very natural because if we talk about like how to identify a species, then very likely we are using some comparison, like because there might be some species that can be confused, like two similar species. If we want to say, okay, how to identify one specific species, that people will often use some negative examples, right, to avoid confusing it with some other species. So with this explicit comparison, like our method FinderCam can correctly identify, okay, the most discriminative trait is a wing pattern. And of course, the blue feather is still a trait, but here FinderCam works specifically for the fine-grained case and highlights the most discriminative trait, and that is the wing pattern. So we also demonstrate that in addition to classification, we can also use FindCam in multimodal scenarios because we can use these bird, for example, this red-winged blackbird, we can use bird and red applet as two classification weights. And similarly, if we only use GradCam, then it highlights the bird very well. But for the red applet, it only has a very weak, very vague activation on the applet. And in comparison, if we create an explicitly contrast between bird and applet, and now FindCam can create a very strong signal on the red applet, and at the same time, very interestingly, it can also exclude red applets from bird. So this tool is like what we used in BioCap to create this explanation map, and it works very well in helping explain like why the model recognizes these species. So with FindCam, we are able to locate and discover biological traits and concepts. And this line of work focusing on discovering biological knowledge from images has been well explored by a broad community. So we have hosted the imageomics workshop at multiple conferences. So in last year, we have hosted in AI 25 as well as NeurIPS 25. So there are a lot of speakers and organizers. They are interested in discovering this biological knowledge from images using AI. And we are also welcoming like more people to join our community. Yeah, so again, let's do a quick summary. So in this work, so in this talk, we introduce how to scale and create data to solve the data scarcity problem. And also we have discussed a way to create more interpretable and explainable modeling. And the next step is really how to advance the scientific discovery. Suppose we have acquired some better models, right? So the question is, how far are we from discovery? So in this talk, we discuss how to transfer raw pixels into some foundation machine learning models, as well as to how to create the saliency explanations. And the next step is we want to know some modeling that is like influenced by context, as well as we want to quantify these concepts. We want to quantify these explanations, and then these two directions can then lead to the discovery of the unknown. So firstly, I'm talking about some future directions. Firstly is context-aware modeling. We know that ecology is actually like study how the animal or how a species fit in the environment. So a species it is affected by the climate, some predators, resources, and other environmental factors. So we need to integrate this information to the modeling of different species, because we know that environment is influencing the phenotype. So for example, we have an image. We know that in GBIF we have an image. We have its geolocation label. We have a time label. But beyond these, we want to incorporate more information, such as the temperature, precipitation, and even some remote sensing features. So this integration can help us better model the underlying reason of different appearances and in their interaction with environments. And this is the first step. Okay, so how do we acquire a context-aware representation? And the second step is how we transfer the explanation to concepts. So why is this necessary? Because if you ask a model, like using one of our previous saliency map methods, like why this image is a red-winged blackbird, it'll very likely tell you, oh, it's because of these pixels makes it a red-winged blackbird. But the question is it because of the shape or color or texture that makes it a red-winged blackbird? By only highlighting the pixels, these questions cannot be answered. Therefore, what we need is a discrete multimodal concepts. We need to explain using natural language that this, for example, like this patch presents features different from other categories because of what? So we need this natural language to help understand like what exactly the trait is. And also, with these discrete concepts, we are able to quantify like how strong this concept is for different images. And subsequently, we can develop the visual genome for, so this is a like concept, like we call it visual genome. So by some visual appearance features, we can identify a species. And later, once we have developed all these different concepts, for example, some features on the head, some features on beak, on tails, on different features, we will be able to connect these concepts to the metadata, to like what time it is and what location the image is captured. And further, we can connect them to more covariates like climate, remote sensing, and local biodiversity, and this whole network will enable us create hypotheses, and these hypotheses can then be verified by scientists. By further, they can develop some experiments, they can develop some verification methods to verify if this hypothesis stands or not. So this is the broad future. And again, so this work, we discussed three aspects of machine learning of AI for science. So we start from data, and we demonstrate that with good data, we can develop good models that have domain-aware representations. And then from these models, we are able to extract the concepts. We can interpret a model, and these concepts can further support discovery. And in return, based on these concepts, they can help us understand, okay, what data do we need if there is a specific task that we need to solve. So it will in turn help us like curate new data, collect new data sets. So together, this loop can help us advance the scientific discovery. So I hope that this is the kind of the broad vision of how to incorporate AI, how to incorporate machine learning models to help scientific discovery. And yeah, thank you very much. So that's the content of this talk. And these are the people I want to thank, that our work is based on these collaborations. And yeah, thank you for listening. And I'm ready for any questions.
Awesome. Thanks a lot for your talk. It's super exciting. Very interesting work. Is there any questions from the rest of the audience? Yeah, go ahead, Kate.
Yeah, this is really great. And I'm glad that you ended with data because I was sort of curious. I have a little bit of familiarity with the databases like GBIF and so forth. And so are you like, is there a preference to, I know that some of those records for observations will also include in the Darwin core metadata, like an extended measurement of fact that includes some environmental parameters that were collected at the same time as the observation sort of thing. So is that a useful source of information for pulling into the models, or are you just sort of like taking it from everywhere, or do you find it more useful to take like a broader like regional data set that incorporates like climate information?
Yeah. So that's really a good question, and this is like an ongoing effort. So we are trying to like use, yeah, just as you mentioned, that we have the geolocation and time labels. So we are trying to use this to probe the available like environmental covariates. For example, like some satellite data that they have the temperature and precipitation for those geolocation time points. And also we are collecting some more like ecological metadata for different species. For example, does this bird migrate? This kind of metadata, we are trying to incorporate this knowledge into modeling. And we want to see like will this model have more comprehensive understanding and will this model enable like more hypothesis proposal?
That's great. Thank you. Really good work.
Thank you.
Great. So maybe I can steal the last question in the last 2 minutes. So earlier in your talk, you were talking about how labels are not informative, right? Or in fact, they can be a little bit misleading in in a lot of cases, right? And so this we've seen in some work providing like a knowledge graph or a graph-like representation, or like a scene graph or a feature graph or somewhere in here has been really helpful. Is this something that you've considered on this side?
Yeah, definitely. That's really a good suggestion. So here we are actually using a very, although we are using some context, like we are collecting information from Wikipedia and we are designing some like format examples, but it's still very straightforward. So yes, just as you suggested, if we can build some scene graphs, like how especially I think especially is useful for cases where there are multiple species in the image, right? There might be an like a bird sitting on a tree. So there is interaction between different species, and I think the knowledge graph, the scene graph you describe will be especially useful for those cases for helping us understand how species interact with each other. Yeah, definitely. And we also know that like currently there's a lot of bias in the data set. For example, like a lot of tree images, they actually labeled as birds because there might be a tiny bird sitting on a tree. But if the image is only labeled as bird, then we are only training the model to say that this is a bird image. So, but the model will kind of discard the major background, the major object of trees. And this is inherently some bias existing in the data set. And what you describe will largely help solve the problem that we can create more kind of interactive descriptions for each image. And I think that'll definitely help the model understand each image better.
Yeah, awesome. Thanks a lot. And that does bring us to the top of the hour. So thank you very much for bringing your work and insights to our meeting today. Really appreciate it, and thank you very much for your presentation. Have a great rest of your day and your week.
Yep. Thank you. Thank you very much for having me.